This is the video lecture for Biotech ENR, and this is a video lecture to be to replace the lectures that I missed on Friday, March 2nd, and also on Monday, March 5th. Where we left off, we had started chapter 8, and so we're going to talk about DNA forensics. And I wanted to thank you as a class for being so gracious to our high school visitors on last Wednesday. You were extremely patient, and I know that that was much of a review of a lot of things that you guys already know. I did want to take out some time for the high schoolers because there were so many of them. And you were very, very helpful. So anyhow, I thank you for that. So we were talking about DNA fingerprinting and talking about different regions of the human genome that can be used as a fingerprint. So we can take really small portions of the genome, not only doing restriction length analysis, which we talked about previously, but we can look at variable number tandem repeats, VNTRs. Sometimes these are called satellite regions. And these are just repeated sequences in the genome. Usually to characterize an individual or an ID an individual, you'll look at about 13 or so of these regions. And each person has some VNTRs that are inherited from mom or from dad. These are also called microsatellite regions. And you can see a microsatellite region here down at the bottom where there's a flanking sequence on either side. The flanking sequence it's not a palindrome of each other, it's just considered a flanking sequence. And then in the microsatellite region, it looks like a broken record where you have TTA multiple times. And in this particular microsatellite, TTA repeats 8 times. And so that individual has 8 repeats on one allele, not both, but just one allele in this microsatellite region. And that was inherited from either mom or dad. Okay. And these short tandem repeats, it's a different type of repeat like a microsatellite. The FBI has chosen 13 of these in its CODIS database in order to identify criminals. Okay, and you can see the 13 different regions here listed out on the 22 autosomes as well as X and Y. And so with these regions, then you can look at how many repeats an individual has for each region, and you can use them to ID the individual. CODIS stands for the Combined DNA Index System, and this is something that happens if you commit a felony, then automatically a blood sample is taken, and then you're entered into the CODIS database. And so in terms of rules of evidence, anywhere in the United States, if somebody wanted to track you for a particular crime, then they could look at these short tandem repeats, have that information readily available, and then after you've committed a felony and you're released from prison, then if they pin you on at a crime scene, then you can be subject to more judicial action. And so there are about 5 million individuals in the CODIS database, people who have committed felonies in the United States. It's getting larger as our prison population is getting larger. And then they can use these 13 regions in order to identify criminals. Okay, so when we prepare a DNA fingerprint, a lot of this is intuitive, and a lot of this we've covered because you know how to run gels and you know how to do comparative analysis. But to collect a specimen, then you have to consider that anything as a source of DNA can also be a source of contamination. So obviously you want to make sure that the individuals collecting the specimen are not contaminating with their own DNA, okay? Because sometimes somebody can have dandruff, their little flakes of skin or whatever can fall into the sample, and then that is a compromised sample. Okay, so they have to wear gloves, use disposable instruments, avoid talking, sneezing, coughing, avoid anything, touching anything that might have DNA, and then air dry the evidence to prevent mold overgrowth. Okay. Sunlight and high temperature can degrade DNA. Bacteria then can also degrade DNA, and then you've got contaminating, degrading DNA, and then moisture, which will allow for mold growth. And so when you get the DNA and you've done your analysis, then it's a comparative process. So you must compare the crime scene DNA with the suspect DNA. And if you can get fresh whole blood, especially leukocytes, then that's going to be the best sample. However, you can retrieve the older DNA and then sort of revive it, rejuvenate it using PCR. So remember, DNA has a half-life of 60,000 years, so a decade is not that long, especially if the DNA has not been exposed to sunlight, moisture, or bacteria. Okay. So then you purify it chemically. Just use some type of detergent. You can use SDS, sodium dodecyl sulfate, or you can mechanically press it out of the cell using something like a French press. And then you want to use PCR to amplify the DNA found in a crime scene so you can have enough quantities to do a real analysis. So the flanking regions for the microsatellites on the CODIS database, those flanking regions are well known, so you can design your primers in order to do PCR through the 13 CODIS regions. So we just do PCR and get multiple copies, and if we do 32 cycles at 100% efficiency, then we get over 1 billion copies of the targeted DNA. And then after we are amplifying our samples via PCR, we separate them using capillary electrophoresis, which is much easier and faster than gel electrophoresis. And then we can allow for the number of repeats of each of the two alleles to be determined. Okay, so here's the results of capillary electrophoresis for different regions. And you can see the different bands for each region corresponding to how many tandem repeats you have. Okay, and so you get this, run this. You've got some duplicates on some of these capillary electrophoresis scans, and then some that will be unique depending on what chromosomes that you've amplified up. And based on that, then you can get, you see the two alleles for each region. Okay, so 2 bands for each region. Okay, so the number of repeats within an SCR is referred to as an allele, just standard molecular biology term. And so say if we have D7S820 STR on chromosome 7, you can see 5 to 16 repeats of GATA. And say you have an individual that with these alleles that have 10 and 15 repeats, then they would have inherited a copy of 10 GATA repeats from one parent and a copy of 15 GATA repeats from the other parent. Okay, and so there are 12 different alleles for this SCR and 78 possible genotypes. So you have 1 in 78 chance of identifying the criminal, which is not good. I mean, we want to not narrow this down to 78. We want to narrow this down to 1 in millions. Okay, so we have to do all 13 of these STR regions in order to do that. Okay, and when we do that, the odds that 2 individuals would have the same STR patterns for all 13 would be over 1 in a billion. So it is a really robust identification method. It's a possibility. Monozygous twins would, if you had a twin, the twin could commit the crime, and DNA evidence could not rule between the two individuals. Okay, and so you compare the evidence from the crime scene to the evidence collected from a known source, from a suspect, and then testing continues only until a difference is found. If no difference is found after you have like a 1 in a billion chance, then the probability of a match is high. Okay, and so here is a mock-up. And we have Jane Doe and Dorothy Smith. And you can see Dorothy Smith right away can be excluded because you look at the first loci tested, D3S1358, and Dorothy Smith is 1518, and the evidence is 1516. The bad news is for Jane Doe, she cannot be excluded because her alleles match up directly with the two stains that were taken from the knife blade. Okay, and so looking at a little history, in 1983, the first reported use of genetic fingerprinting was done in the UK in the Narborough village murders. Okay, this was sexual assault and murder of a girl in the UK, and the prime suspect's DNA did not match that of the crime scene. And so they looked at a big sampling of suspects, and it turns out that none of the DNA profiles matched the crime scene, but they did match the crime scene to a friend of the suspect, and the friend of the suspect had given the sample. And so when the friend had an alibi, I don't know why you would do that. That's if a friend ever asks you to give a sample for a crime scene, don't do it. But when the friend had an alibi, then they were able to then remand the suspect, get an actual sample, and then match that up to the crime scene. Okay. So all you want to do is you want to take your specimen, add detergent, incubate it overnight, centrifuge and discard the supernatant, then add extraction detergent without ditheo. I can't remember what DTT is. I think it's on the next slide. Okay, but then you incubate it, save the supernatant containing female DNA. I am not sure about this slide. I'm really not exactly. I think this might be for paternity. I'm not quite sure. Okay, let me look at this. Okay, let's think about that slide later. That just appeared in random. So DNA evidence was first used in the US in 1987. And it has been used to overturn convictions. Now that we have DNA evidence and DNA can be preserved for a long period of time, then a lot of convictions have been overturned. Okay. And here is just a sample between two people there. Okay. And then you have to look at how reliable is DNA testing. Eyewitness is actually, a really strong standard, and law and culpability is established by eyewitness testimony. But it turns out that in the case of the Forest Hills rapist, Victor Lopez DNA was a match, and he actually was tried for the sexual assault of 3 women. They reported that the assailant was a black man. Lopez was not black, but his DNA was a match to the crime scene. Okay. Also, In terms of terrorism and looking at, identification of remains and natural disasters, there's um, new technologies that have been developed because a lot of times, when a bomb blows up or when, say, a flood occurs, then you need new technologies because the DNA can be degraded. Okay. So in the World Trade Center and the 9/11 attack, they used DNA techniques to identify the remains of victims. And so because of the debris, the heat, and the moisture, then microbial growth decompose the remains, and they had to analyze via DNA a bunch of tissue samples from nearly 3,000 individuals. Okay. And because people lost their loved ones and they wanted answers very quickly, then the forensic scientists involved in the World Trade Center terrorism attack had to profile DNA very quickly. And then if somebody was missing a relative, they would submit a sample from that relative, or say if they were a brother or sister of the relative, they could submit their own sample, and then they could do DNA profiling that way. Okay. And so if you were missing a loved one at that particular time, then you could give a sample of your DNA. You file a missing persons report and you provide a cheek cell swab. And also sometimes they would take personal items that might have cells from that individual. Okay. And this spawned the development of new software programs. They use STR, which is a nuclear genomic technique. So you need actual like blood or skin or a cheek cell sample. They use mitochondrial DNA because in some cases they only had like the hair shaft. And so hair DNA is mitochondrial, it's not nuclear. And they did SNP analysis. The tsunami that hit Indonesia and South Asia, the mortality was over 225,000. And they use mitochondrial and Y chromosome STRs, which obviously is only good on males. Mitochondrial works on anybody. And so they had identified 800 victims that way. Okay. In terms of rules of evidence, DNA fingerprinting has to meet legal standards. Okay. So there are the relevancy test, the Fry standard, which deals with the scientists that did the DNA work, the Coppolino standard, the Mark standard, and the Daubert standard. Okay, and Daubert and Fry actually deal with the qualifications of the scientists. I'm not exactly sure about Coppolino and Mark's. Okay, in terms of rules of evidence, let me go back to that. That's kind of bugging me. I think I might review that in class because I want to know more about these standards. They are talked about more in your textbook. I don't happen to have my textbook with me while I'm doing this recording because I'm at home, but I will go back to that and review that in class. Okay. The Nicole Brown Simpson and Ronald Goldman murders. This is a very high profile case because it involved O.J. Simpson. Happened in 1995. Okay. They collected 45 samples for DNA analysis, including samples from the victims and the suspect, blood drops at the crime scene, suspect's home, and in the suspect's car. Obviously, the suspect here was O.J. Simpson. Okay. And in pretrial hearings, they announced that the DNA collected at the crime scene matched that of O.J. Simpson. Okay, there's a lot of evidence that has matched O.J. Simpson to that particular crime. Okay. Defense lawyers went about attacking all of the methods involved in DNA collection, labeling, and testing. and there was a possibility that the evidence was tainted. And you don't really have to prove that the evidence is tainted. You just have to provide some type of doubt in that particular instance that the evidence was correctly handled, and they were successful in raising that doubt. Okay, but in that case, DNA evidence was not effective because at the end of the trial, O.J. Simpson was declared not guilty, although he, in a later trial, was declared guilty of um, a wrongful death suit. And so it's a very, very interesting case. But again, the DNA evidence, because of missteps in terms of the Los Angeles Police Department in handling the evidence, the DNA evidence was ineffective. So we have to look at what are the sources of contamination. Okay, so you have to record, who had the evidence when. That's called chain of custody. And the access to the evidence must be controlled. Obviously, the defense attorneys, prosecuting attorneys, everybody is going to want the evidence. So it's very, very important to establish a chain of custody just in case the evidence is altered. Okay, and then there are defined standards of laboratory practice that are defined by the FBI, defined by local authorities in order to prevent DNA damage during the analysis. And this is all science. And so when you talk about the rules of DNA evidence and you're talking about DNA evidence to a jury, you're talking to mostly non-scientists. You've got 12 jurors and very few of them are going to be scientists. So it has to make logical sense to the jury. And the statistics can be confusing. Say if you say 1 in 50 billion, that may lead somebody to think, oh, there's a possibility that the DNA evidence is wrong because there is a 1 chance. So 50 billion, there aren't 50 billion people in the world. That would have to be explained very carefully. Another type of analysis that's done is familial relationships. So paternity testing is very, very common. If a woman has multiple partners and becomes pregnant, then you need to be able to establish who the father is, especially if there's a paternity suit that's being involved. And so you want to analyze samples from the child as well as the adults involved and see, is this man the father of the child? Interestingly enough, now there's maternity testing for women who denied that they were actually pregnant and carried the child. So there have been some maternity suits as well, which seems very, very heinous that somebody would do that, but there is a precedent that's been established for that. Okay, and so here we have short tandem repeats from 3 different regions, actually 4 different regions. One is X chromosome, one is Y chromosome. So you've got the mother on top, child in the middle, and the father on the bottom. Okay, and you can see from these alleles, you can see that the child is carrying alleles from the mom and the dad. Here's allele 13. The child is homozygous in 13. It got 13 from mom and dad. Here's the allele 28 and 29. Got 28 from mom and 29 from dad. And then here's allele 15 and 16. It got 16 from dad and 15 from mom. So this does establish paternity. Again, you would want, this is only for STRs. You would want to do all 13 in order to establish paternity. And it is possible to draw fetal cells from the amniotic fluid to test for paternity. It's not recommended because this type of amniocentesis does cause miscarriage. There's a 1 in 300 chance that in an amniocentesis that a miscarriage could occur. So, but what you would do is you would just withdraw a sample of amniotic fluid. This can cause premature labor. And that's where a miscarriage is a problem. You take these cells from the amniotic fluid, which are presumably from the child, and then you cultivate those cells to get more of them, and then you have enough DNA from the baby to be able to test. Also, you can use mitochondrial DNA for familial relationships. Okay, if you can't use RFLP or STR because you can't get a nuclear sample, say if your material is hair, bones, or teeth, then you can't get nuclear DNA from that, so you'd have to do mitochondrial DNA. Then you can only establish maternity. You can't establish paternity because mitochondria are obviously inherited from mother only. And the changes are very, very low. So this is a high fidelity type of test. Okay. And so if we look at mitochondrial DNA patterns for different individuals, you can see that the mitochondrial DNA from female is always passed down. So if you've got a female who gives birth to a female, then all the grandchildren will have the same mitochondrial DNA. Here, same thing. Female gives birth to a female, all the same DNA. Now, if a female gives birth to a male, then the mitochondrial DNA dead ends because the female that then mates with that male is going to pass on her mitochondrial DNA. There's also Y chromosome analysis, which is only good in males, passed directly from father to son. And it's good if you've got a male child and you're trying to establish paternity for that male child. Okay. There's also non-human DNA analysis to confirm the authenticity of a particular species. Ginseng is very, very important. It's got a $3 million market in the US alone. It's actually a pretty small market, but still, you want to establish, is it North American ginseng or Asian ginseng? Asian ginseng boosts synergy, the American ginseng calms nerves, and the American variety is more valuable. So you want to make sure that when you get ginseng to calm your nerves, that you're getting the right variety. And the way that you do that is a genetic test because the two varieties of ginseng are genetically different. Okay. Cabernet Sauvignon grapes, if you want to trace these back to France, to the ancestral Cabernet Sauvignon grapes, then you have to do DNA evidence. You have to find that the DNA is either Cabernet Franc or Sauvignon Blanc, and then you can establish pedigree. If you have hybrid grapes, if you haven't bred them correctly, you've introduced a new variety, then after the DNA test, then that wine cannot be designated as Cabernet Sauvignon. You can use this for poaching. Okay, here, they traced a bear in a den, and witnesses reported that a hunter discharged gun into a den, which is highly illegal. You can't kill bears in their dens. Okay, and the bear's premolars were removed at the registration site, and that confirmed the identity of the bear, and then that was matched to blood that had spattered in the den. Okay, and that then showed that the hunter was hunting illegally. And then DNA tagging, any object, any inanimate object can be tagged with DNA. Okay, you just put a little tag on it, it's got a DNA sample that's preserved. And so you can authenticate different materials just by the DNA pattern. They do this for Super Bowl footballs, paraphernalia, medals from the Sydney Summer Olympic Games to help fight fraud and verify authenticity for people that trade such things. Okay, and that concludes the video lecture, this at least for chapter 8. Let me see, I'm going to go on to chapter 9. I'm actually going to pause the video here. You won't notice the pause, but I'm going to pause the video as I pull up chapter 9. Chapter 9 is on bioremediation. And so bioremediation is just using microorganisms primarily and other organisms in order to clean up contaminated waste sites. So we're going to define bioremediation in a little bit greater detail. Talk about some of the basic techniques that are used in bioremediation, some of the different approaches, how effective they are, how much they cost. We'll look at how to approach a different site and how we would come up with strategies for cleanup. Talk about the application of genetically engineered strains to clean up the environment. We have to be very careful with any type of GMO released to the environment, because as the contamination goes away, then we want to make sure that the genetically engineered organism dies off. And then we'll talk about some case studies. We'll look at oil spills as well as large groundwater spills. And we'll talk about some of the challenges and what we expect for the future of bioremediation. So what is bioremediation? It's basically biodegradation. So if we use any type of living organism, it can be bacteria, it can be fungus, it can be plants, to degrade chemical compounds, that's biodegradation. And if we extend this to bioremediation, this is just the process of cleaning up these sites using living organisms to degrade hazardous materials into less toxic substances. And that's very important. There are some instances in bioremediation where, unfortunately, they've taken hazardous materials and made them more hazardous, and we'll talk about that throughout this chapter. So the ultimate objective is to make sure that you're not getting rid of contaminants only, but that you're reducing them into something that is less toxic. Why is it important? Because environmental chemicals are bad for us. Okay, they influence our genetics. A lot of them are damaging to DNA and can cause mutational effects over generations. Short and long-term exposure can lead to things like cancers, chronic illness, and inheritable diseases. And if you look at how much hazardous materials we produce in the United States, it's huge. And we're smarter than we were before. We're curtailing specific contaminants in the environment, but we're still disposing hazardous materials. And in 1980, the U.S. Congress started what was called the Superfund program. This was a part of the United States Environmental Protection Agency. And what Superfund does is it basically counteracts careless and even negligent practices of chemical dumping and storage. There are a lot of sites where, especially during war efforts, during World War II, and the Korean conflict and the Cold War, there was a lot of careless contamination of sites in the name of developing bigger and better weapons. Okay. And so our purpose of the Superfund site is to locate the most egregious sites in terms of the amount of toxins and clean them up. Okay. And 1 in 5 Americans lives near a polluted site treated by the EPA. We do. We live near Iron Mountain Mine, which is 9 miles outside of Reading, and it is an EPA Superfund site. It's also the location of the lowest measured natural pH in the world. And EPA has cleaned up greater than 700 sites in the 25 years after Superfund, but there's still over 200,000 sites that need to be remediated. And so to look at these costs, it's greater than what we would expect the US government's budget per year. So it's going to take a long time to clean up these sites, and some of these sites will just not be cleaned up. Okay. To understand further what's going on, there's the Environmental Genome Project, and that is to look at the interactions between genes and environmental chemicals, not only in human health, but also to look at genes that are involved in detoxification, say in microbes and other organisms. And then finally, identifying SNPs that might be indicators of environmental impacts on humans' health. Some people that have specific SNPs or have specific mutations may be more susceptible to contamination than others. And we need to generate genomic data, and then we can look at who has these SNPs via epidemiology, and then we can understand how the environment contributes to disease risk and how specific diseases are influenced by environmental chemicals. These would be long-term diseases like Alzheimer's disease, Parkinson's disease, autism spectrum disorder, ADHD, and we can understand why these diseases are approaching epidemic proportions and what toxins in the environment may be impacting that. So why would we use bioremediation? Okay, it turns out that if we use biological methods for organisms that have adapted over time to degrade contaminants, they will degrade the pollutants into something harmless. Okay, most organisms are not going to degrade something into something more toxic because that would just kill them off. So if they've adapted over time, then usually they will degrade the contaminants to things like carbon dioxide, water, simple organic molecules, and say chloride ion. The processes are generally cleaner. They can be conducted in situ at the site of pollution. And we can identify then what needs to be cleaned up. Well, almost everything has, pollution affects water, air, sediment. It should not say oil, it should say soil. I don't know why it says oil. Soil, water, air, most common, okay? Each presents its own complexities for cleanup because of the site dependency of bioremediation. And you see pollutants enter the environment in many ways. Sometimes it's unintentional, like a tanker spill or a truck accident. Sometimes it's just release of pollutants that happens naturally over time. Some manufacturing facilities, back when there were not regulations on pollution, would just mainly dump their pollutants into a body of water or dump them directly on the ground where they would end up in groundwater. Okay, so you need to look at what's the location of the spill, the accident, the amount of chemicals released, and the duration of the spill. And that's going to impact how the environment's affected and whether you're going to have an exposure to human health. Okay, so we look at some leaky tanks. There's a surface tank that's leaking. It can contaminate the air because of vapors, say from the chemicals. It will contaminate the surface soil and then ultimately go down below the water table into the groundwater. Subsurface tanks will also leak in the groundwater. And then we have to look at the impacts downstream. Are we creating a hazard here with surface water with this leaking chemical tank? Are we creating hazard with groundwater with this subsurface tank? And ultimately this groundwater is going to be tapped by wells to provide drinking water and other types of water for these homes. Okay, so we have not only the upstream chemicals, but we also have to look at the downstream exposures and how do we remediate these chemicals before the exposures are affected. Okay, there's lots of different chemicals in the environment. There's carcinogens, mutagens that cause DNA mutation, teratogens that specifically cause birth defects, skin rashes, and those that poison plant and animal life. So we're not just protecting human health, but we're also protecting the entire environment because humans rely upon the environment. Okay, and here's a rundown of some of the different chemicals that are used. A lot of remediation has been developed for chlorinated organics. So things that have chloride in them, say like, I get my arrow to work here. Where's my arrow? I don't see my arrow. Oh well, you just can read it. My arrow seems to have frozen. So perchloroethylene, tetrachloroethylene, which is PCE, trichloroethylene, which is TCE, and trichloroethane. These are degreasing and dry cleaning agents. There was a lot of degreasing that was associated with the aircraft industry. So you find a lot of these contaminated sites at Air Force bases. Polychlorinated biphenyls. Carbon tetrachloride is another one that's not listed on here. Explosives like trinitrotoluene, and then trace metals. So if we look at the fundamentals of cleanup reactions, the reaction chemistry is usually pretty well characterized. But we're looking at aerobic metabolism and anaerobic metabolism. And in aerobic metabolism, usually you have to provide oxygen in some way, shape, or form. In anaerobic conditions, you may not need oxygen, but you also are going to look at supplying another terminal electron acceptor because the organisms will need an electron acceptor, and if that's not available in the environment, then it needs to be provided. Okay, this involves redox reactions. So oxidation would be removal of one or more electrons. Reduction is the additional of one or more electrons. And you'll see contaminants oxidized or reduced depending on the nature of the condition. Okay, so here's just a redox pair. You've seen this before, oxidation and reduction. Okay, and then we have aerobic biodegradation. And you'll see in aerobic biodegradation, the terminal electron acceptor is oxygen. So this organic chemical like benzene is converted to carbon dioxide, water, and then biomass as the bacteria increase. This is not balanced because we're using just an example hypothetical chemical. Here, if we have something like toluene, toluene is more preferably, it will go by aerobic conditions, but here the terminal electron acceptor is nitrate. And so we're making CO2, nitrogen gas, and hydrogen and water. And in this anaerobic condition, then we would have to provide the nitrate, or nitrate would already have to be in the environment for this reaction to occur. Okay, so when we look at metabolizing microbes, we need to understand the physiology of these microbes and how they've adapted to their particular environment to clean it up. Okay, some strategies involve combination of aerobic and anaerobic bacteria to fully decontaminate, especially if you're looking at something like tetrachloroethylene. That will only, the first chlorine will off of tetrachloroethylene will only be reduced by anaerobic bacteria, and then the rest can go aerobic. So you have to be very crafty in the bioremediation strategy that you use. Okay, and if we look at a contaminant plume, let's say we've got soil and groundwater, and then we've got a petroleum spill right over here. Okay, this is the source. And usually, when the source is contaminated, the first thing that's used is oxygen. Okay, so as this contaminant plume moves throughout, it has already degraded all the oxygen, so only at the very plume front would you find oxygen. So out here we have an aerobic zone in the white, and the contaminant, say, benzene again, is being reduced aerobically. Okay. In the denitrifying zone, there's only nitrate and there is no oxygen. So nitrate reducing anaerobes are found in this zone. After nitrate is depleted, then the next zone would be iron reducing. Okay. And iron is reduced from iron 3 to iron 2. by anaerobic bacteria. After all the iron is reduced, then in the next adjacent zone you would have sulfate-reducing organisms. And after all the sulfate is reduced to hydrogen sulfide gas, then you would have methanogenesis, where the organisms are converted to methane by the action of carbon dioxide. This doesn't show a real good reaction. Usually methanogenesis will reduce carbon dioxide, but the reaction chemistry is a little too complex to show on this very simple slide. So we need to remember those zones. Furthest away from the contaminant, you'll have a natural aerobic zone, then denitrifying, then iron reducing, then sulfate reducing, then finally methanogenic. So you should know those zones, should know the basic reactions, and you should know them in sequence as in proximity to the contamination source. Okay, one of the easiest things to do is just use the microbes that are already in the polluted site, in the soil or the groundwater or the surface water. Since the contaminant has been there for a while, then the microbes have adapted over time and are using, to some extent, the contaminant as a food source. Okay, so you can use this indigenous rather than trying to inject microbes directly into the surface, which would be much more expensive. Okay, so sometimes you can grow and study in the lab and then release them back into the treatment environment in large numbers. Other times you can do the whole thing in situ, where you just increase the numbers just by stimulating the activity. Okay. Pseudomonas, which is usually Pseudomonas aeruginosa and E. coli, are really, really good for degrading oil spills, but you need them in larger numbers than they will occur naturally in the soil. Okay, and there's always a quest to identify new metabolizing microorganisms. That's always an active area of research. Okay, and when you get new contaminants like arsenic, then you will find organisms that have adapted to metabolize this. There's a bacterium in Mono Lake that metabolizes arsenic and incorporates it in its DNA. So it basically isolates the arsenic and puts it directly in the biomass of the bacteria. Okay, you can also use algae and fungi. Phanerochaete chrysosporium and Phanerochaete sordida are associated with wood chemicals, and they will degrade creosote, pentachlorophenol, and other pollutants where bacteria cannot degrade these. Okay. Asbestos and heavy metal degrading fungi include Fusarium oxysporum and Mortella hyalene. And fungi are always, if you have like a large compost pile where you're degrading waste, where you have sewage or sludge at a solid waste facility or wastewater treatment plant, you'll always see fungal growth. And fungus can degrade polychlorinated biphenyls, PCBs, that were used quite frequently in manufacturing as well as in electrical transformers. And we're finding that fungi will degrade those better than bacteria. Okay, and I think I will conclude my lecture here. And this will not be, chapters 8 and 9 are not on exam 2, they're on exam 3. So you're studying up to chapter 7 for exam 2. We'll cover this in more detail as we do the review for the exam on Wednesday, March 7th.